Carlos, welcome. Are you ready now? Okay.
Absolutely.
So let me say just a few words about you, Carlos. So Carlos is Carlos Ribeiro. Thank you very much, because this is the second time that you kindly accepted to participate. So thanks a million. And Carlos worked at, let me see whether I pronounce this properly, Champalimau Foundation in Lisbon, Portugal. And he's interested in the neural mechanisms of nutrition, especially how the neural circuits control nutrition and decisions, and how these decisions can affect aging and reproduction. So thank you very much, and all yours, Carlos.
Okay, great. Thanks for unmuting. Yes. Sorry for the computer problems before. It's really a pleasure to be here. It's always, when I get the invitation of DrosAfrica, it's always one of the things I need to say yes, because it's really a pleasure to talk in front of this audience, which is really a fantastic testament to what science can do and bringing us together and showing us the future. So I'm going to be sharing with you a bit our work on how we are trying to understand how the brain makes decisions, more specifically how the brain tries to make decisions about what to eat. And in the presentation, I put a bit more emphasis at the beginning on more general concepts and approaches, and at the end, I'm going to have a bit more of a newer story we have been putting together. But I prefer to have a really well understood first part than to really have to go to the end. So if you have questions, just feel free to ask them at the end, and we can go into that. So now this works again. So first of all, let me thank my team. I'm very, very blessed and lucky to be able to work with this fantastic group of people with very different backgrounds, from physicists to microbiologists to geneticists, and we're all working together to try to understand how the brain makes decisions. I'd also like to highlight here our collaborators, right? Science is very interdisciplinary, and we're lucky to count on our collaborators, which allow us to bring new dimensions, new techniques, new ideas into our work, and to go beyond what we could normally do. And also, I would like to thank our funders, the grant agencies which fund our work and which keep us and our flies well fed, with a special emphasis here on the Champalimau Foundation, who is our main funder and who is also hosting us at the Champalimau Center for the Online. So what we would like to understand is how the brain makes decisions about eating or not eating. But in contrast to what most neuroscientists want to understand, which is how the brain decides to eat or not to eat, we think that actually what the brain should really be trying, or what the animal is really trying to optimize, is what type of food to eat. So if you look at food, right, we immediately appreciate that the nutrient content of food can be very different, right? So this, for example, some foods have more carbohydrates, some foods have vitamins, some foods have more proteins, and some foods have different types of fats. And we know that different nutrients have very different impacts on the fitness and the behavior of the animal. And sometimes, or often actually, different nutrients even have contradictory and opposing results. So the animal really needs to be able to reconstitute nutritionally optimal diet by choosing to eat from different foods, so it has enough nutrients to optimize its reproductive and its lifespan. And from all these nutrients, most obviously of the research, especially focused with the bias from our Western world, is focused on understanding how energy and fat actually affects behavior and physiology, because there's this obsession with obesity. But there's a nutrient which we are very interested in, and for which we don't know so much about how our brain regulates the homeostatic intake of it, and that's protein. So proteins are made up of 20 amino acids. All 20 are important for making proteins, but 10 of these are special because they're essential, meaning that we cannot, or we can only very poorly synthesize them. And actually, interestingly, the same amino acids which are essential for humans are also essential for Drosophila. And this is really interesting for me as a neuroscientist, because that means that the animal really needs to, the only way the animal has to have access to these essential amino acids is to eat them, right? We're also not good at storing amino acids. What happens if we run out of amino acids is we start chewing up our muscles, which is not really a very smart strategy, right? So that means that protein and amino acid intake needs to be very tightly regulated by the brain. So the brain needs to really find out when when the animal is lacking out amino acids and make sure it eats them. But a simple strategy where the animal would just eat and overeat amino acids is also not really optimal, because work of the last 15 years has clearly shown that an overingestion of proteins and amino acids leads to a strong decrease in lifespan and also health span. And this in all animals, again, from invertebrates to mammalians to humans. And in humans, obviously, this is based on epidemiological studies, but I think the data are pretty clear. So for me, that's really interesting, right? So so we need to eat enough amino acids so that we can make protein, but we shouldn't eat too much amino acids, because then our life is going to be shortened. And how the brain really calculates this trade-off is something we would like to understand, and which I would actually argue is at the basis of understanding what does it mean to eat right, and how does our brain and our body encode eating right and make sure that we eat right. So conceptually, this is what we would like to understand, but how do we bring this into the lab, right? How do we study this in flies? So now I'm going to show you how we conceptualize this now in the lab, okay? So this is a simple experiment where we have flies which are getting the choice to eat from two different food options. One food option is yeast. Yeast is the main amino acid source from the fly. It's kind of the steak of the fly, or the chicken, or whatever you want to see as protein source. And the other option is carbohydrates, in this case, pure sucrose, right? And so these are the two options which we offer our flies to eat. And we have three different types of flies. Flies which have been kept on a full diet, flies which have been amino acid deprived, and flies which have been carbohydrate deprived. And what you see is that the amino acid deprived animals will drastically and significantly increase the intake from the yeast, so from the amino acid source. And you get the same effect if you give them pure amino acids, but they will not increase the intake from carbohydrates, whereas the carbohydrate deprived animals will not increase the intake of yeast, but they will significantly increase the intake of carbohydrates. So this means that the animal knows that it's lacking certain nutrients and can then bias its choice at its intake to then actually compensate for the lack of its amino acids. Flies are very good at this. Insects in general are very good at this, but also rodents and also humans do this. So if you take people and put them for 2 weeks on a low protein diet and check what they will eat, they will generally overeat proteins in the washout phase compared to people which have been kept on a high protein diet, which means that we are also able likely to compensate and for a lack of proteins and amino acids by overeating proteins homeostatically. And I will propose that this is really at the center of understanding nutrition, right? Because obviously the brain is a central regulator of nutrition. And if we understand how the brain regulates our choices of food and how it tries to make sure that we have the nutrients we need, we might be understanding what are the needs of the animal, what are the rules underlying these needs, and then also how they change the brain so that kind of we make the appropriate decisions. But today I would like to focus a bit on what can we understand and what can we learn about the brain by studying nutrition, okay? By studying nutrient homeostasis. So if we now take a very cartoonish view of the brain, right? So I'm a trained cell biologist, so I like to break things into simple mechanisms, right? So you can think about the brain as taking in external sensory stimuli like taste and olfaction, processing these, then this information going to higher order complex brain regions, central brain regions, and these then making computations and decisions of how the motor neurons will then at the end produce a behavioral output, right? This is called a sensory motor arc, and this is traditionally where most of the work actually in sensory biology has gone into over the last 20 years, understanding how sensory inputs are transformed in specific motor outputs. But I think always, and more recently with a more profound interest, people have known that these are not fixed reactions, right? And that there are so-called internal state, internal variables like hunger, thirst, tiredness, which will modify how we use and perceive sensory information and to what type of motor outputs that will lead. And we think that by understanding what these internal states are and how these internal states affect the brain and how the brain uses sensory perception, we can understand really how higher order cognitive computations are done. And so what is our strategy to identify the mechanisms by which our brain does this, right? So we believe that we need to work in an interdisciplinary approach to really understand that. We need to understand the neuronal circuits, the neurons, how they take in this information or change. We need to understand what are the molecules, what are the genes, what is the genetic basis, the nutrient sensing mechanisms, which shape neuronal circuit function and adapt circuit function so that we make appropriate decisions. We need to understand how the nutrients and the microbiome too actually affect circuit function. And we need to do that using quantitative behavioral assays because it's really by looking at the details of the behavior that we can have a deep mechanistic understanding of how the brain alters sensory processing to change behavior. We also need to ask why that happens, right? So we always look at what are the consequences of perturbing these mechanisms. And we look here mostly at aging and health, and in flies as a health output, we look mostly at reproduction as a readout of physiological function. And more recently, we've become interested in trying to understand how metabolism and physiology in general, especially specialized organ physiology, interact with all these factors to shape neuronal circuit function. And I would propose that it's really at the intersection of all these different topics that we can get a deep mechanistic understanding of how nutrients affect brain function and behavior. And ultimately, how nutrition is encoded in the brain. Obviously, such a complex and ambitious interdisciplinary approach is best done in a powerful model organism. And this is really where the fly shines, because it has complex enough behaviors, it has complex enough physiological regulations, while allowing us to do precise mechanistic perturbations of molecular and neuronal function, and also to do that in an unbiased, hypothesis-free approach so that we can really discover new mechanisms by which this happens, right? But let me walk you a little bit how we are trying to really get at how internal states affect brain function, right? And here really a very important tool, which we were lucky to collaborate with Matt Piper and Linda Partridge at UCL, was a development of a chemically defined diet, okay? So most studies in flies use yeast. As I said, yeast is the main protein, main amino acid source of the fly. It's also what the fly is really specialized to eat. But the problem is that yeast is also a very complex nutrient source, right? It also has carbohydrates, fats, vitamins, lipids, etc., etc., etc. So when you remove yeast from the diet, you don't really know what you really manipulate in terms of nutrients in the animal. And so the development of a fully chemically defined medium, which we call the living medium, which means that it's really completely chemically defined, allows us now to replace yeast by specific chemicals which are defined at nutritional level, and now to not anymore just put in and remove yeast, but really remove and manipulate single amino acids, lipids, vitamins, and methyl ions. So it gives us full access to the nutritional variables which control the behavior and the physiology of the animal. This has been an extremely powerful system to really look at how specific nutrients affect the physiology of the animal. This is actually something also which very, very few animals systems have, and which is one of the strengths for doing nutritional studies in the fly. So what are examples of non-nutritional variables which can also affect the fly behavior? So one of them is the microbiome. So I think over the last 10 years, a growing realization has happened in the field that the gut microbes, so the bacteria, the fungi, the viruses we have in our gut, but also around our body, somehow shape brain function and how the brain works and how we make decisions and how we make behavior. And we have uncovered that the fly microbiomes of flies also have gut microbes strongly change what the fly wants to eat. So here I'm showing you a very simple experiment where we have germ-free flies. So these are flies which have no bacteria in their gut, and these are fully fed flies and flies where we remove isoleucine from the diet, and we are measuring how much yeast these flies need. And as you're seeing in flies which do not have a gut microbiome, they have a very strong increase in protein intake, because they want to compensate for the lack of dietary isoleucine. But when we put two specific bacteria from the gut microbiome into these flies, Acetobacter pomorum and Lactobacillus plantarum, these two bacteria strongly suppress the drive of the flies to eat protein, even if they lack isoleucine. And this is a strong evidence that the gut microbiome has a strong impact on fly behavior, and that's something we're trying to understand. And we already know that these bacteria do not change the behavior by giving the animal amino acids, so they're doing something else, and that's an active field of research in the lab. As I said, we also showed that peripheral organs, so the physiology, the metabolism of organs like, for example, germline in the female, talk to the brain and tell the brain what they need so that the brain will eat enough. But another very important variable which changes what animals want to eat, and this is what I'm going to dive a bit more deeply into, is reproductive state. So in females, if a female is virgin or mated, therefore producing offspring, will have a strong impact on what they want to eat. We know that kind of anecdotally from obviously women, and there's also some strong data showing taste reprogramming in pregnant women, but this is also something which has very well been documented in insects, especially flies. And so here is one of the examples showing that. So these are 4 types of flies. Flies which have not been protein deprived, so they've been kept for 0 days on a diet lacking amino acids, or flies which have been deprived of proteins for 3 days, and we have virgin and we have mated females. And as you see, in virgin females, they will slightly increase protein intake after 3 days of protein deprivation, but this effect is not very strong. Whereas if we do now the protein deprivation in mated females, so females which are producing eggs, there is a synergistic effect, interaction between reproductive state and nutrient deprivation. And these females now strongly increase the intake of protein, meaning that females which are mated will now strongly prefer to eat yeast and also other nutrients like salt. My former graduate student Samuel Walker, as you can see from his tea drinking proper Brit, looked into what are the circuit mechanisms which control that. And what he found is that this is all regulated by a specific set of neurons which react to the sex peptide. So what we know is that virgin and mated females behave very different, right? So virgin females are receptive, they don't make eggs, they retain their eggs. And also, as I said, they do not like proteins. And mated females, they are unreceptive, they don't want to mate with males, they lay a lot of eggs, and they like a lot of proteins. And this is induced by the sex peptide. The sex peptide is a small peptide produced by the male, and which the male injects into the female and reprograms its behaviors from being virgin-like to being mated-like. And this is classic work from the Kubi and Wolfner laboratories, which show that you can inject the sex peptide into a virgin female without them ever encountering a male, and these females will now all behave like females. And in my postdoc with Barry Dixon, I cloned the sex peptide receptor, which is a GPCR, which is expressed in most neurons in the female brain, and which receives the signal from the sex peptide and changes the behavior of the female. And to make a long story short, there is a very small set of neurons which have projections in the vaginal region of the female, which detect the presence of the sex peptide and bring this information up to the brain of the female and will tell the female that she is now mated or not mated. So there is only 4 neurons which are required to bring this mating information to the brain of the female. And interestingly, we can trick the female to think that she's mated and make her eat protein as if she would be pregnant, right? So here what we are doing is we are silencing these neurons, which I just showed you, bring in the sex peptide information. These are these flies here. And normally virgin females do not eat a lot of protein, whereas mated females eat a lot of protein. These are the control females. And when we now silence these neurons in the virgin, this virgin will eat as much protein as if she would be mated, okay? So we have all these factors here: nutritional, nutrient sensing mechanisms, mating sensing, the interaction of different organs in the animal, which all will set the internal state of the animal and somehow change how the brain reacts to sensory information and what it will eat. Okay, and how can we look at how these changes in behavior act on the brain? So what we have been doing there is really using how the fly eats. Okay, so if you look at the fly feeding behavior, and I'm going to slow this video down a little bit, the fly uses this really very stereotyped proboscis extension retraction cycle to eat. And really, we can measure food intake and food choice in flies using more low-tech and more high-tech approaches. And we choose which one we want to eat, depending if we want to do a lot of assays or if we want to really look precisely into the animal. The easy way of, or the high throughput way of doing that is by using the red-blue choice assay. So here you mix one type of food like protein with blue color. You mix the other type of food like sugar with red color. You let the fly eat for 2 hours, and then you just look if their abdomen is blue or red. If yeast is mixed with blue color, that means, and the fly is blue, it means that the flies preferred the yeast. And if the flies are red and the sugar was mixed with a red color, you know that the flies prefer to eat red, and you can calculate an index for the preference based on the ratio of blue to red flies. But the way actually we decided to bring that a little bit more high-tech was to use touch screen technology. So can we measure how the fly eats by measuring how it interacts with food by using the way that our touch screens work? And so the idea was to use capacitance sensing using the same sensors which are in your cell phone, which cost around €2 a piece, to measure when the fly is touching the food and therefore count just how many times the fly touches the food and reconstruct how the fly eats. This led to the development of the Flypad setup. So this is the Flypad. We put here an arena where we have 2 of these touch sensors, one on which we put yeast, the other sensor on which we put sucrose. We put one fly into this arena, we close the lid, we connect this to the computer, and then the computer will automatically and high throughput measure what the fly is eating. And here is what you're seeing here is that on top is the measurement of what the fly is eating from the capacitance sensor, synced down here to the fly eating from the food. And if we now correlate the numbers of sips, so that numbers of times the fly has touched the food with how much the flies actually ate, what you can see is that there's a very, very nice linear relationship between food intake and the numbers of sips, okay? And so now I would really like to end by really looking a little bit into the brain of the fly while it's eating, okay? Because now we know that different internal states affect the brain and that somehow the brain then changes the behavior of the animal. But how can we find out what's going on in the brain? So how can we find out what changes in the brain when nutrient appetites change, right? And so here what we're using is we're using calcium imaging. Okay, so the principle is the following. So if you are lucky like me to have very skilled postdocs, what they can do, or graduate students, they can open the cuticle of the brain, over the brain, so the cuticle of the head, in a way that the fly remains alive. And then you can glue this fly under an objective of a 2-photon microscope while the fly is still alive, and while we can also bring food onto the mouth of the fly. And now importantly, we are expressing a fluorescent sensor in specific neurons of the fly brain, which is called GCAMP. And GCAMP has a very nice property. It was engineered to not glow when it is not bound to calcium and to glow when there is calcium bound. And what's important to know is normally when when a neuron has action potential, it will release calcium in the neuron, and that will lead to GCAMP actually glowing, okay? So what we're doing here is we will be looking at the fly brain. More specifically, we're going to be looking at taste neurons, which are going to go to the fly brain. So we're going to look at taste information going to the fly brain, and we're going to look at how these neurons fire when the fly is eating proteins, okay? So here, again, we're going to be looking at the activity of these neurons when the fly is eating, and here we're going to look at the activity of that quantified. And what you see when yeast is put on the tongue, these neurons fire, and we can measure this very nicely here with the calcium activity increasing. Okay, again. Okay, And so now what we can start doing is we can do that now for multiple and different neurons in the brain and start understanding, do these neurons now fire differently when the female is pregnant or is not pregnant, or when the female is lacking specific nutrients or not. And to really finish here, a very talented postdoc in the lab, Daniel Münch, has asked, can we do that in a more systematic way? Do we need to do that neuron by neuron? Because the adult fly brain has around 200,000 neurons, and there's not enough time of a postdoc or a student to do that for every single neuron. And so what Daniel has done is he has now expressed GCAM in all neurons in the brain and done GCAM imaging in all neurons at the same time. So here again is this poor little fly here under the objective, under the 2-photon objective, and it's going to be eating different types of foods while we're going to image its brain while it's eating. And here you're seeing now a 3D rendering of the ventral part of the Drosophila brain while it's eating protein. And we are scanning slowly in Z direction, okay? And you see that now we can see a lot of different brain regions here in the fly brain firing and having activity when the fly is eating. And just because I'm going to spare you all the analysis which we have done, which you can read up in our latest bioRxiv paper, already looking at this very superficial level, you can see that there are very big differences between fully fed flies and protein-deprived flies, which we're now analyzing and looking at in a more detailed way. And this is really one of the approaches we have been using to really dissect how the brain controls food intake and different nutritional states, and how different nutritional states impact the behavior of the animal. This is extremely relevant in terms of approaches, because we always like to think about flies as models for human physiology and diseases, but we shouldn't forget that insects are a main factor when thinking about human well-being. Obviously, they're vectors of disease like dengue and malaria, something I don't have to tell you, but also they are one of the main reasons why we lose actually on full harvest. But I think that we shouldn't forget that ultimately, at least for me, this, being a scientist also means understanding the beauty which surrounds us. And here I would really like to leave you with a slide of what I find a very beautiful scene you can find in the wild, which are male butterflies drinking the tears of a turtle. And the reason the male butterflies do that is because they want to get the salt from the tears of this butterfly, so they can give this salt to the female when the male would mate with the female, so that the female will be able to produce a lot of eggs. And this is called the nuptial gift. Obviously, the males are doing that in their own interest, but it's because of this nutritional requirement of reproduction that we get to appreciate and also understand this beautiful picture here, which we find in nature. This is my great team. I'm eternally grateful for being able to work with them. And I also would like to invite you to join our virtual seminar series, which we have on brain-body interactions every Monday, where we explore this fascinating developing theme of how the brain interacts with the body and how the body interacts with the brain, and how we can study the brain looking at these things, and how many diseases and phenomena of brain function are shaped by this interaction. Thank you very much for listening to me.
Okay, Carlos, thank you very much. That was amazing. It is incredible how much you can know about the fly. So
absolutely, right? Imagine we would fund it accordingly, right?
So we have time for just one question, which is from Pablo again, and Pablo asks, does the Drosophila cat secrete brain intestinal peptides?
Sorry, does the gut secrete
brain intestinal peptides?
Yes, obviously, yeah. So I mean, obviously there is a lot of crosstalk between the gut and the brain, and the other way around too. And this is beautiful work from many people, especially Irene Miguel Aliaga, right, which has shown that there are neurons, right, which bring information to the brain from the gut. But there's also many peptides which are being secreted there, and we have actually some really nice stories looking into that now, where we have ideas about how signals from the gut might also be controlling physiology and behavior area. But we also know that the brain directly reads out the nutrient state of the animal, so we can molecularly kind of trick the neurons to think that there's not enough amino acids, and then actually the fly will also eat more protein. So it's a whole integrated information collection from the periphery, from peripheral organs, from the gut, from the microbiome, but also autonomous nutrient sensing, which ends up shaping the behavior of the animal.
Okay, thank you very much. In any case, guys, if you have more questions, you can post them in Slack, and then Carlos will be able to answer them.